So I'm from the Cancer Discovery Hub, which is part of the National Cancer Center of Singapore. The CDH is a primary functional multi-omic core facilities, and our current director is Dr. Jason Chan. We collaborate with clinicians at the NCCS as well as other institutions. We aim to provide state-of-the-art research tools. We do bulk-seq, we have single cells. We're also involved in spatials, Visium. We are now actively involved in the BGI stomics because it's a high definitions. We have an MGI sequencer. And once we have all the paperwork done, hopefully we might have an onshore sequencer park at CDH as well. And in addition to that, we do the whole 9 yards. We make tumor model creation, PDXs, organoids, cell line out of all these different cancers. We are part of a nationwide consortiums. The STCCs are the one whereby we are involved in making organoids, cell lines, PDXs. In addition, with our collaborators, we have this Symphony, which is a lymphoma consortium, which we are part of it. Tetrad, which is the nationwide leukemia, as well as our very own Starlight program whereby we look at rare cancers. And we hope to use all these different tools and platforms in order to get enough preclinical trial data so that we can complement our clinical trials and support advanced point of care at the NCCS. So for today, I'm going to talk about the cell-free DNA work that we've done. Cell-free DNA, cfDNAs are short DNA fragment that we are interested in. Typically, they tend to be found in low quantities in body fluid like blood, urine. Normal individuals, we all have cell-free DNAs in our bloods. They're from turnovers from the white blood cells. In cancer patients, we have a proportion anything from less than 1% to 90%. What we term circulating tumor DNAs. They're from the turnovers of the tumors. So potentially we can get genetic information, epigenetic informations from this cell-free DNA. And the cell-free DNA advantage over tissue biopsy include non-invasive. You only have to take some blood from the patient rather than doing a surgery to remove a tissue biopsies. As a result, it's safer, potentially less costly, and it's more reflective of the tumor heterogeneity. Imagine if you have tumors in different part of the body. If you only take tissue biopsy from one side, you only get the profile of that side. So the one that you can gather from the cell-free DNA potentially can give you more a better reflection of the tumor heterogeneity. So you can imagine that if you have single liquid biopsy, this is a hypothetical patient whereby depending on which way you get the cell-free DNA, it can provide early cancer screening, early diagnosis, and molecular profiling, and as well as detection of residual disease once you have any treatment going on. But the most important that interests us is the serial liquid biopsies, whereby you can get a lot of interesting data that are useful, like monitoring responses to certain drugs, as well as monitoring clonal evolution. The cancer that we're going to look at is a rare cancer called gastrointestinal stroma tumor, GIST. It is the most common soft tissue sarcomas. GIST is defined by the activating oncogenic mutation in KIT genes. The small minorities are the PDGFRA, and there are other small minority drivers. So concentrating on the KIT, the primary KIT mutation that induced the active kinase activity is due to mutation in either exon 9 or 11. So for the past more than 20 years now, ever since the discovery of imatinib, which originally worked in BCR-ABL in CML, they found that it's also useful against KIT and PDGFRA. So it was immediately used for GIST. So GIST originally turned out to be a very fatal disease. I think now it's much more manageable than before. But however, as you know, once you use the drugs, you're going to get relapse. And many of the mutations are found in exon 13, 14, and 17. But again, it's like a war between, you know, the drug makers and the cancer. And now there's a whole range of TKIs that you can use if you come across any imatinib resistant mutation. Now I'm going to talk about three different kind of platform: one for variant and two for DNA metallome. So for this cohort, we have cell-free DNA collected from the plasma, 64 of them from 46 GIST patient. For looking at variant, at that time we are using the Archer liquidplex. Originally, it has a 28 gene panel. We include one more gene because it is we've shown in publication that it's important in GIST. So this is the distribution of the different. As you can see, majority of them tend to be. Oh, anyway, just looking for the laser pointer. Anyway, you can see that on the left, majority of them tend to have the KIT mutation. The small number have PDGFRA. When it comes to the disease stage, small number of them tend to be localized. Majority of them tend to be metastatic or metastatic with evidence of progression. So what are the kind of mutations that we are able to detect using this platform? So first of all, many of the data that we have of known KIT mutation from the corresponding primary tumors are based on Sanger. So we only have either Sanger-seq for the KIT or the PGFR because they are the one that are druggable. And to be honest, they are the one that define this disease. So we also have, on the NGS platform, obviously you can pick up anything that is on the panel. And we can see that here that we can only detect them in the metastatic with disease progression. Unfortunately, we cannot detect the KIT mutations in any of the cell-free DNA from the localized and non-disease progression metastatic. But for these additional mutation which we find in TP53, SETD2, KRAS, and IDH2, majority of them we can still find in metastatic, but we can't confirm because we don't know whether this mutation existed or not on the original tumor because we do not do an NGS on it. So now we come to the serial plasma samples. So this is a retrospective studies, and then the aim is to detect changes in mutation profile, KIT or other genes at different diseases and treatment stage. So for this patient, the known mutations in the red box is the exon 11, the typical activating mutation. And on the y-axis, you have the mutant allele frequency. And in this case, we have 3 cell-free DNA samples taken at different weeks. And on the x-axis, you have one row for what are the drugs that are used. And on the bottom, you have the clinical status. You have the CT scan at the different weeks. And you can see that initially we cannot detect the KIT mutation in the plasma, but over time as the disease, SD stands for stable disease, PD progressive disease. As the disease progresses, the tumor get bigger, and we know that once it get bigger, you can get more cell-free DNA in the plasma. And that's where we detect the original exon 11, and we also detect the imatinib-resistant exon 17 begin to appear. But the interesting thing in this case is that you even get the oncogenic PI3K kinase mutation. This is a very well-known E545K. It's a very pathogenic variant, and we are able to detect at week 26. So this patient had a radiotherapy due to stomach lesion in between 26 and 42, and all of a sudden that basically killed off the KIT clones that have the KIT. We don't detect cell-free DNA from KIT anymore. Instead, we see a huge explosion of the PI3K. So it appears that it's one of the very rare cases of GIST whereby they completely switch their dependency from KIT and now rely on PI3 kinase. So this is a much more complex, but just bear in mind that original in the red boxes, exon 11, exon 17, the green, and the purple, the purple. You can see that in the 0 week, we can only detect the original green exon 11. As the patient take up this drug avapritinib and then followed by imatinib, you can see that progressively the green start to drop on week 4, and then you can start to see the exon 17, the imatinib resistant gene begin to go up. And by week 10 and 16, when they are responsive to reparatinib, which is another TKI, you can see that once they become responsive, you don't detect the cell-free DNA. And once the disease began to progress, you begin to see the original exon 11. And in fact, you see the yellow one, which has not been reported. You can see a new exon 13 TKI resistant mutation arises again. And you have the similar things here. Initially, you can detect the original exon 11 mutation at week 0. Over time, when they become responsive and the patient is stable, you can see that you are not able to detect any of the mutations. So once the disease begins to progress, that's where you start to see the exon 17 mutation. So which means that when they switch it to a different TKIs, you can see that it's responsive again. So all these serial responsive serial plasma allow us to correlate the detection of mutations as well as correlate with the response from the patient. So one thing that we decided to look for is that we know that simply looking at variant may not be enough to in trying to use cell-free DNA for analysis. So we began to look more into DNA metallome. So just to give you an example, so typically there are more than 28 million CpG dinucleotides in the human genome, and 7% of them are found in CpG islands that represented something like 25,000 CpG islands. So for a given cancer, the example here is a particular kind of lymphoma. You can see that the number of differentially methylated regions between normal and the cancer is basically in the thousands. And when you compare it to SNV, the variant that you can find for a given cancer, exemplified by prostate cancer here, they say for a given cancer, average number of genes with recurrent hits are relatively low. So, 1,000 versus tens. So if you're trying to look for circulating tumor DNA, it's like looking for a needle in a haystack. So if you have thousands rather than tens to look for, then your chances of identifying circulating tumor is far better. So we decided to give it a test. We did this. This is based on a publication in Nature in 2018. It's called the CF-MIDIP-6. It stands for cell-free methylated DNA by immunoprecipitation. They claim that they can do it with as little as 5 nanogram, but to be honest, I think in our experience, you need at least 20 nanogram to really get a good signal. It based on using well-established anti-methylated cytosine antibody to isolate out cell-free DNA fragment that have the CpG islands that are methylated. So we did that with 8 metastatic samples, and we can see that you can get 340 differentially methylated regions that distinguish the GIST cell-free DNA from normal ones. And you can see from the PCA plot that you get very nice clustering of the GIST samples, and then separation from the normal. And among these 8 GIST samples, if we were to use just the KIT mutation, we can only detect 3 out of 8. So here we can detect 8 out of 8. So that gives us confidence that the way to go is, in addition to variant, we should be looking at the DNA metallome. Going forward, we're now switching more to the EM-seq method, which is similar to sodium biosulfide. The EM-seq method basically uses two enzymes, TET2, which protect the methylated cytosine and prevent ApoBac that convert the unmethylated cytosine to uracil, and the end product is the same as bisulfite. The EM-seq method appears to be better because it provides higher yield, less PCRs, consistent global methylation levels, and so forth. And the panel that we're using is from TWIST, which basically target about 4 million CpGs. This is just an exercise whereby we share the 3 different cell lines into about 150 base pair just to mimic the cell-free DNAs. We use 5 nanogram, 10 nanogram, 20 nanogram. The whole idea is to look at the QC efficiency. At least using cell line, it looks like 5 nanograms pretty. You can get pretty decent result. And these are the coverages. Basically saying that using 2 different algorithms, the average number of methylated is similar for different inputs. And I think, to be honest, I think even though this shows that as little as 5 nanogram, in reality, for cell-free DNA, you definitely need, again, at least anything from 20 nanogram to 30 nanogram minimum to get a good signal. So to summarize, the various NGS platforms that we use for low input, I've shown you the variants. We've shown that in order to improve detection, you definitely need variants in metallome, cell-free DNA variants in metallome, and then it's important to know all the different cancer-specific variants in DNA metallome in your cancer before you start using cell-free DNA as a cancer biomarkers. And we are now switching more towards the Twist EM6 because the EM6 provide base resolution data. Because the CF-MIDIP6 is just simply based on antibody capturing a CPG-rich area, but you can't tell where are the methylated CPGs from that. And finally, to end, acknowledgement, I'd like to acknowledge my mentor and supervisor, Professor Tay Bing Tien, for his support, and also Cancer Discovery Hub. I'd like to thank the director, Dr. Jason Chan, for his support as well as other members of the of CDH, the clinician at NCC, people from the TBT lab, and as well as our industrial partner. And on the bottom right, we have the disclosure of the project funding as well. Thank you very much for your attention.